Hello, good morning, all of you, and welcome to this PhD webinar series, which is organized by Microbiologist Society of India. Today, we have organized a lecture of Dr. Sarika Girdhari, and the topic is studies on production and process of optimization of microbial tannase by using tannin rich agro residue. First, I will introduce herself. She is qualified MSc and PhD and working as an assistant professor in the Department of Biotechnology, Shiva Chhatrapati College, Aurangabad, and she is working as IQSA member. She has published 3 research articles and she also published 3 books, and she has presented 9 paper in different types of seminars, and she has also completed 1 research project, and the number of citations are 7 for her research article, and H index is 1, and also she got a first prize in oral presentation in international conference, that is ICMTBT 2080, which is organized by DY Patil Vidyapit Pune in association with Microbiologist Society India. So, ma'am, you can start your lecture.
Yes, ma'am, sure. But I am unable to share the screen right now.
What is problem, ma'am? Sarika, ma'am?
It's one message that hosts disable participant screen sharing. Hello.
Your PPT.
Yes, ma'am, I am unable to share that because one message is there, host disable participant screen sharing.
Post disable. And you got the message, ma'am, this?
Yes, on my screen, this message was there, host disable participant screen sharing. डिसेबल किया है शेयर करने के लिए पीपीटी.
Okay. Let's see.
Ask the host to give the right permissions.
The host should enable the screen sharing for the participant.
Wait a minute, ma'am, I will see. Wait a minute, ma'am. Wait.
Okay, ma'am.
Mute their video and sound. Mute your sound and video.
Ma'am, still I'm unable to share my PPT. Meanwhile, shall I start in the background?
Within few minutes, the problem will be solved, ma'am. Can you share? Can you start the lecture?
Yeah. I will start in the background. No problem. So, good morning, all of you.
Ma'am,
Dr. Sarika Girdhari from Shiv Chhatrapati College. First of all, I would like to say thank you to the Microbiology Society of India for giving me the opportunity to present my research work. the journey of this research was started in the year 2012. And the topic of my research is studies on production and process optimization of microbial tannase by using tannin-rich residues. So I have carried out this research work under the guidance of respectable Professor Swati Peshwe from Government Institute of Science, Aurangabad. So the purpose behind to choose this topic was to develop a process which will be environmental friendly and economically feasible and which will be fruitful. Yes, yes.
Share your PPT, ma'am. आपको co-host किया है, ma'am.
Yes, ma'am.
How you can share the PPT, ma'am? Please.
Yes, ma'am.
Okay. Okay. It is visible to all of you.
Okay, thank you, ma'am.
Okay.
So the purpose behind this work is to develop a process which is economically feasible, which should be environmental friendly, and which gives the fruitful result to the society. After studying the extensive literature, we found that there are so many problems behind the occurrence of the tannin in the nature. So these tannins are occur in many edible fruits and vegetables, which are considered nutritionally undesirable because they form the complex with the proteins and digestive enzymes and cause the reduction in the nutritional value of the food. Therefore, it exert the anti-nutritional effect by the protein and tannin complex and reducing its availability of the food nitrogen. In animals, especially in the ruminants, these tannins have a toxic effect which can reduce the feed intake and lowers the nutritional digestibility and the protein availability. Again, the another problem related with this tannin is which is used into the tanning process, and the wastewater which is generated from the leather industry contains the tannin which is highly polluting in nature. This tannin also forms the insoluble precipitate in beer, wine, and coffee flavored soft drinks, and therefore the various types of the fruit juices show the haziness. So to resolve this problem, one solution is there, that is the application of the enzyme tannase. Tannase, which is also called as tannin acyl hydrolase, it is an inducible and hydrolytic enzyme, and it catalyzes the hydrolysis of ester bond and dipeptide bond of hydrolyzable tannin. Its activity was found to be sequential, whereby cleavage of the dipeptide bond occur first and followed by the hydrolysis of the ester bond. So when we observe the reaction, it acts on the tannic acid and produces the gallic acid and the glucose. This slide shows us the structure of the tannin. It is nothing but the polyphenolic compound of the varying molecular weight, and it is a polymer of gallic acid and the glucose. Then it is a fourth most abundant plant constituent after the cellulose, hemicellulose, and lignin. So what are the application of this enzyme tannase? This is used into the wastewater treatment of the leather industry. It is used in food, beverage, cosmetic, and chemical industry. We can use this tannase in clarifying as a clarifying agent in some fruit juices and in cold drinks. Then in manufacturing of instant tea, we can use this tannase. In the production of the gallic acid, also it is useful, as well as it is useful into the poultry feed also. Nowadays, it is observed that this enzyme is also helpful for the detection of the colon cancer. So not only the enzyme, but its product, that is gallic acid, has the tremendous application. This gallic acid is used in the pharmaceutical as an antibacterial, antiviral, and analgesic agent. It can be used in the cosmetics as an antioxidant. In a photographic developer, we can use this gallic acid. Then in manufacturing of ordinary writing inks and dyes, in tannery industry for the homogenization of the tannin, and in production of the pyrogallol and gallic acid esters, we can use this gallic acid. So objective of my research work was to find out the efficient tannase producing microorganism, as well as to find out the tannin rich agro residues. And using the combination of these two, production of the microbial tannase using the process optimization method and study its application. So to optimize the process for the enhanced production of the microbial tannase, we have a particular roadmap with us. The number of soil samples we have collected for the isolation of the organism, which are tannase producers. So 13.63% actinomycetal isolate we have isolated, then the 22.72% fungal isolate and 15.90 bacterial isolates we have isolated for this tannase production. And the screening was done accordingly. Total 52 fungal isolates were obtained, and amongst that 52 fungal isolate, 30 fungal isolates were found to be tannase producer. Then different 50 bacterial isolates we have isolated, and out of 50 bacterial isolate, 21 bacterial isolates were found to be tannase producer. In the case of actinomycetes, 30 different actinomycetal isolates we have isolated, and amongst 30 actinomycetal isolates, 18 isolates were tannase producer. So the primary screening of the tannase producing microbial isolate was carried out first of all with the help of the plate method. And you can see here the isolates which are showing the clear zone of tanninolysis. We can call them as a tannase producer. So tannase producing fungal isolate here you can observe, which are showing the zone of tanninolysis. These are the actinomycetal isolates showing the zone of tanninolysis, and same thing with respect to the bacterial isolates. After primary screening, secondary screening of tannase producer microorganism was carried out by using the Mondal's assay. For this Mondal assay, we have used the 0.5 ml of tannic acid, which is nothing but the 1% in the 0.2 molar acetate buffer of the pH file, and 0.5 ml of the enzyme extract was added. Then this mixture was incubated for 30 minutes. After the enzymatic reaction, this is further continued by addition of the 1 ml of the bovine serum albumin for the precipitation of the unhydrolyzed tannic acid. And again, this tube was centrifuged at 10,000 RPM for 10 minutes at the 4 degrees Celsius, and precipitate was dissolved in 2 ml of SDS triethanolamine solution. And the absorbance was measured at 530 nanometer after addition of the 1 ml of 0.13 molar of FeCl3 reagent. After carrying out this secondary screening, the tannase unit was defined. So 1 unit of the tannase is defined as the amount of enzyme which is able to hydrolyze 1 micromole of ester linkage of the tannic acid in 1 minute under the specific condition. So with this secondary screening, we have selected some potent tannase producer organism from fungal, bacterial, and actinomycete isolates. So you can see here this SKF7, SKF11, SKF12 fungal isolate, SKA2, SKA9, and SKA1 were the actinomycetal isolate, and SKB4, SKB2, and SKB3 was the bacterial isolate, which considered as a potent tannase producer organism on the basis of the secondary screening. After performing the primary screening and the secondary screening, location of the enzyme was found, whether it is an intracellular or extracellular enzyme. So literature survey tells that the tannase is an extracellular enzyme, and we have also checked it, and the results show that the enzyme was the extracellular in all the isolates which we have considered as a potent tannase producer. Then after isolation of the organism, which are efficient tannase producer, we turn to the collection of the agro residues and their phytochemical screening. So different types of the agro residues we have used, and phytochemical screening were carried out. So the solvent extraction was carried out for the measuring the tannin content of these different agro residues. For this purpose, we have used the dried powder of the various agro residues with the 80% methanol in 1:4 ratio. and that mixture was kept on shaker for 72 hours. And then this mixture was filtered, and filtrate was subject to the centrifugation. Then this filtrate was concentrated by evaporation to yield a gummy concentrate till it reduced to get the 1/4 of its original value. And this residual content was mixed with the methanol and then again suitably diluted to check the availability of the tannin content in that agro residues. So the total phenol content estimation is first of all necessary before you are going to measure the hydrolyzable tannin content. So total phenol content was measured by the Follin-Dennis method. For this purpose, we have used the 1 ml of extracted tannin, and it was mixed with the 1.5 ml of Epsi reagent. Then after 5 minutes, the 4 ml of 20% sodium carbonate solution was added, and volume was made up to the 10 ml with distilled water. Then this mixture was incubated at room temperature for 30 minutes, and absorbance was read at the 738 nanometer. So when the tannin-like compounds reduces the phosphotungstone molybdic acid in alkaline solution, it produces the highly colored blue solution. So this this Follin-Dennis method relies on the transfer of the electrons in alkaline medium from phenolic compound to phosphomolybdic acid complex to form the blue complex. And this blue color complex we have determined spectroscopically, that is the measuring the absorbance at the 738 nanometer. Again, after carrying out the total phenolic content, we carried out the estimation of the hydrolyzable tannin content in that agro residues. And for this purpose, again, we have used the Mondal's assay. So these are the 31 different agro residues which we have used for our study. Different types of the agro residues like the leaves, then the seeds, and some fruits we have also used. And amongst these 31 different isolates, we found that the Azadirachta indica leaves contain the higher amount of the total phenol as well as the higher amount of the hydrolyzable tannin content. As well as not only this Azadirachta indica, but the custard apple leaves and the almond leaves also shows the higher content of the total phenol and the hydrolyzable tannin content. Therefore, for the further study, we have selected these 3 different agro residues, and then we have tried the permutation and combination of the potential microbial isolate with this tannin-rich agro residues. So different combination we have performed with that potent tannase producing isolate and the agro residues. And when we see this graph, we can easily observe that the combination of this isolate SKA1 with the Azadirachta indica leaves shows the promising enzyme activity, higher enzyme activity. So amongst the actinomycetal cultures, we have used this SKA1 with the combination of the Azadirachta indica leaves, and this SKB2 with the Prunus dulcis leaves, and this SKF7 with this Prunus dulcis leaves again for the study. So this slide shows us the microscopic observation of the fungal isolates. So this Aspergillus fumigatus, Aspergillus carbonaris, we have isolated. Then the SK1 isolates, which we have used for our study. This slide shows us the electron micrograph of that actinomycetes isolates. Then after this, the genotypic analysis of this actinomycete SKA1 culture was carried out. Then it was found that based on nucleotide homology and phylogenetic analysis, the selected actinomycetal culture, that is SKA1, was identified as Streptomyces variabilis strain and found to be homologous to BI99. And this sequence was submitted to Gene Bank with the gene accession number KY214403, and it was published on the 23rd May 2017. After selecting the correct combination of the agro residues and potent tannase producer, we have optimized the physical parameters and chemical parameters for the enhanced production of the enzyme. So for the optimization, we have selected certain physical parameters like the inoculum age, inoculum size, incubation period, incubation temperature, and the pH. Then for that SKF7, we have used the 6 different medium. Then the concentration of agro residues varied from 1% to 10%. Then the chemical parameters like the carbon source and nitrogen source, we have studied different carbon sources we have tried for the enhanced enzyme production. Then different nitrogen source also, and amongst that different carbon sources, tannic acid was found to support the maximum enzyme production. The obvious reason is that the tannic acid serve as an inducer for this enzyme production. And amongst the different nitrogen source, the yeast extract was found to support the maximum enzyme activity. Again, the same thing with respect to the bacterial cultures we have carried out, that is the optimization of physical and chemical parameters. For this, we have used the 5 different medium and different physical parameters. For this, we have used the different carbon sources as well as the nitrogen sources. Again, here we got the same result, that is tannic acid shows the promising enzyme activity when we use it as a carbon source, and yeast extract show the promising enzyme activity when we use as a nitrogen source. Then the different physical parameters and chemical parameters also optimized with respect to the actinomycetyl isolate, that is SKA1. And we have used the 5 different medium for this also. With this, amongst the different carbon sources, surprisingly, we find that the starch support the maximum enzyme production, and among different nitrogen sources, the casein found to support the maximum enzyme production. So this is the table which summarizes the process parameter, and they're all physical and chemical parameters. Then again, we have studied the induction and reparation pattern with respect to the tannic acid, gallic acid, and the glucose with the help of the extensive literature survey. So it was found that the gallic acid did not induce the tannase production. And this is because of the end product inhibition also. But the tannic acid support the induction of the tannase production. In case of the fungal isolate, it can induces the production up to 1%. In case of the bacterial isolate, it can induce the production of the tannase up to 2%. And again, in case of actinomycetyl isolate, it induces the tannase production up to 1%. Then the selection of the potent tannase producer for the further studies. Among these 3 different isolate, we have chosen the actinomycetyl isolate because the extensive literature survey revealed that the studies on these actinomycetes other than bacteria and fungi are fragmentary and limited. Therefore, the further study was carried out using this actinomycetyl isolate SKA1. Then we move towards the optimization of the media with the help of the statistical method. So this statistical method for the purpose we have used, that is the optimization of the media component for maximum production of the tannase. For that, we choose the selection of the basal production medium. Then the optimization of the carbon source, nitrogen source, and optimization of the trace element was carried out. Then why we have chosen the statistical design to optimize the media, because there are so many advantages of this statistical design. This statistical experimental designs help us to improve the product yield. Then it reduces the process variability. Then it gives the closer confirmation of the output response, as well as it reduces the development time also. For this optimization study, we have used the Plackett-Berman design, path of steepest ascent experiment, and the central composite design. So Plackett-Berman design is the statistical design which is used to reflect the relative importance of the various factors. Path of the steepest ascent experiment is used to move rapidly toward the neighborhood of the optimum response, and the central composite design is used for the determination of the optimum values of the individual factors. So with the help of the Plackett-Berman design, we got this particular design, and we have used for that purpose the Minitab software. So that software gives us the design, which shows that the variable and its level, low level and the high level with respect to starch, glycerol, tannin, and other substances. So the Minitab software gives us the 12 different trials, and amongst the 12 different trials, the trial number 4 shows the promising enzyme activity, that is the 35.27 units per ml activity we got, and the predicted activity was 35.58. So we have chosen this combination, that is the trial number 4, for the further study. So depending upon the calculation of the p-value, that is the confidence level not less than the 95%, we got that the starch, glycerol, and KNO3 shows the p-value less than 0.05. So these factors consider as important factors for the production of the enzyme. And that Minitab software provide this Pareto chart for the for knowing what are the important factors which affect on the enzyme activity. So by this slide, we can see that the important factors are glycerol, starch, and KNO3, which shows the value less than 0.05. That is the confidence level not less than the 95%. So Pareto chart directly showed that the most important factors determining the enzyme activity were starch, glycerol, and KNO3, while other factors have insignificant influence on the enzyme production. Then the path of steepest ascent experiment was carried out to move towards the neighborhood of the optimum level. For that purpose, we got this particular design from that Minitab software, and amongst the different 8 runs, the 6th type of the run shows us the promising enzyme activity when agro residues were mixed at the 4% level. With the help of the central composite, we have carried out the real value, that is the -1 and +1, and the values which we have taken for the further experiment: starch 1.4%, glycerol 0.37%, and KNO3 0.025%. For central composite design, we have chosen the Design Expert software. And that Design Expert software gives this particular type of the experimental design, which shows the 20 different types of the run. So after studying this 20 different types of the run, after running that experiment in the 20 different flasks, we observed that the run number 1, that is the flask number 1, showing the promising enzyme activity, that is 23.51 unit per ml. Then the analysis of variance for this response surface quadratic model was carried out, and it was observed that the model which is showing the value p-value less than 0.05 was considered as a significant. Then it shows the f-value. Then the low f-value can shows us that is that model was the significant, and there is the independent variables have the variation in between them. So the R square, that is the value of the determination coefficient, we got the 97.13%. So it means that there is 97.13% variability in the independent variables. And the low value of the CV, that is the percent CV, coefficient of variation, that is 8.99%, shows that there is the precision between the different types of the variants. So we got this type of the graph, linear pattern of this graph shows that the normality in the error term, that is there were no any problem in the data. The model which we have used for the optimization of the media was authentic. Then for this model gives us the 3D response curves. So this model shows that these types of the elliptical nature of the graph. So this elliptical nature of the graph shows that there is the correlation between the different types of the independent variables. So here we can see that if this graph is a circular type, it shows that there is the problem regarding that model, and we cannot use this model for the study. But this elliptical graph shows us the model is the authentic, and the this model is significant also. So after carrying out this CCD, we got the result. In that result, the response surface methodology gives us the result in terms of the enzyme unit. So we got the 23.5 units per ml, and the predicted value is 22.21. So our experimental value is higher than that of the predicted value. After carrying out this optimization, we have taken the two different media, that is the original media and the optimized media. So this is the media, which is the original media is starch casein medium, and this is the new media in which the variation is there after the statistical optimization. So this is the optimized media. So with the initial media, we got the enzyme activity 11 units per ml. But with the help of the optimized media, we got the enzyme activity 23.51 units per ml. So there is the twofold increase into the enzyme activity when we use the optimized media by the statistical optimization. After this statistical optimization, strain improvement study was carried out. So this strain improvement study was carried out by two methods, that is the chemical mutagenesis and the physical mutagenesis. For chemical mutagenesis, we have used the ethidium bromide and sodium azide, and for physical mutagenesis, the UV irradiation method was used. So UV irradiation method, we have taken the spore suspension, and it was exposed to the UV radiation, that is 254 nanometer. And after incubation, the colonies above the LD50 we have selected, and they are considered as a mutant. And we have chosen that colonies for the measuring the tannase activity. So this table shows us the exposure time, number of colonies survive, and this is the percent survival. So percent survival curve was used to determine the which colonies we have to use for the further study. So the colonies above the LD50 values, the colony which remain viable after 36 seconds, we have chosen for the further studies. This table shows us that the different types of the strain we have taken. For that purpose, we have taken the wild type and the UV mutant strain. And among different UV mutant strain, the strain UM5 shows the promising enzyme activity. Then the sodium azide treatment was given. Then sodium azide was not considered as an effective chemical mutagen, because after incubation, it is observed that the percent survival of each plate was above 50%. Then the chemical, another chemical use, another method which we have used is the ethidium bromide. So the spore suspension of that Streptomyces variabilis exposed to the different concentration of the ethidium bromide, and after incubation, the colonies after above LD50 dose were considered as a mutant and studied for the post-mutational enzyme activity. So this is the percent survival curve with the ethidium bromide, and the number of colonies we have selected for the further studies. So ethidium bromide concentration vary from 0 microgram to the 7 microgram. And amongst these different types of the strain, the mutant strain ETBRM7 was found to show the promising enzyme activity, that is the 23.5 units per ml. So after carrying out this strain improvement technique, the ethidium bromide mutant strain ETBRM7 exhibited the enzyme activity comparable with that wild type strain. It was observed, but this activity was exactly equal to the wild type strain. This is probably because the mutation was not significant for the enzyme production. Therefore, the only mutant strain UM5 was selected for the further experiment as an enzyme we have selected for the further experiment, which shows that the increase in the enzyme activity by 52%. Then after strain improvement, the purification of the enzyme was carried out. We have purified this enzyme at the different stage with the help of the ammonium sulfate treatment, then with the help of the dialysis, and with the help of the using DEAE cellulose column chromatography. So with the help of the 60% ammonium sulfate precipitation, there is 5.92-fold increase into the into the purification, and with the help of the DEA cellulose, there is 22.67 purification fold we got. So this is the elution profile of the tannase. Here we got the two peak, and the two different peak. There is the presence of the proteins, but at the second peak there is the activity of the enzyme we got. Then the molecular mass determination was carried out using the SDS page, and we found that the one major band was observed, which was 150 kilodalton, and the second is 66.4 kilodalton. And most of the reported purified tannase has a total molecular mass in the range of 168 to 310 kilodalton. So literature survey revealed that the tannase have generally high molecular weight, and they are multimeric form. Then after this study, we move toward the different types of the parameter study, like the effect of temperature, pH, and effect of reaction time, effect of substrate concentration, and effect of different metal ions, as well as the additives. So with the help of the wild strain and the mutant strains, we have carried out the experiment, which is showing the effect of temperature on the enzyme activity. This graph shows us that this green color graph shows that this is the enzyme activity of the wild strain, this is the enzyme activity of the improved strain, and it was observed that the enzyme activity of the improved strain was found to be increased at the temperature of 40 degrees Celsius also. And when we checked its thermal stability, it was observed that residual activity was found to be the 95.32% in case of the improved strain when we expose it to the 40%. And this improved strain enzyme was also stable in the range of the temperature of 40 to 60 degrees Celsius. So this is one advantage for because what happened when the different types of the operations are carried out with respect to that tannase enzyme, they are carried out at the high temperature. So this enzyme definitely can withstand with the operations which use the higher temperature. Then the different pH we have checked for determining the stability of the enzyme. So starting from pH 3 to pH 8, we exposed that particular enzyme for the different pH, and it was observed that the wild strain shows the enzyme activity 58.78%. The enzyme activity was retained at the 5 pH, as well as the improved strain shows the 67.25% of the enzyme activity at pH 5. Then we can also study the pH stability of the tannase. So we observed that the improved strain enzyme was able to withstand with the pH which range from 4 to the 8, and it shows the promising activity, that is the 40.5% residual enzyme activity at the pH 8 also. Then it shows the 45.29% residual enzyme activity at 7 pH also, and higher activity at the pH 5. Then the effect of the substrate concentration was carried out with respect to the that enzyme, and effect of the incubation time or the effect of the reaction time was carried out. So it was observed that with the 30 minutes, the enzyme shows the activity above the 25 units per ml, and With the incubation period of the 50 minutes, the improved enzyme shows the enzyme activity more than 15 units per ml. Then the effect of substrate concentration was carried out, and the different substrate concentration starting from 0.25% to 2% used for this study. And it was observed that if there is increase into the substrate concentration above 1.25%, there is no further increase into the enzyme activity. And the optimum substrate concentration for this is 1.25% for the wild type strain as well as the improved strain also. Then the effect of different metal ions was carried out, and it was observed that in presence of the HgCl2, the enzyme activity of both the wild strain and the improved strain get totally inhibited. Then the AgCl, in presence of AgCl, 70% of the activity was inhibited, and in presence of the ZnSO4 also the activity get inhibited. So this is because of that, as this AgCl and the Zn metal ions may act on the thiol group of that particular enzyme. Then the reason why this HgCl2 is inhibitory, because this HgCl2 inhibits the different types of the amino acids like the histidine and the tryptophan. As it reacts with the histidine and tryptophan, this shows us the inhibitory activity on the enzyme. Then the kinetic parameter study was carried out. So it was observed that the Km value for the wild type strain was 0.11 into 10 raised to minus 4 millimolar, and improved type strain is 0.33 into 10 raised to minus 6 millimolar. So decrease in the Km for the improved strain indicates that more enzyme substrate affinity as compared to the wild type, while the Vmax is also found to have the increase on the strain improvement. Therefore, it was concluded that the strain improvement has improved the enzyme efficiency. After this different types of the parameter study, we have carried out the immobilization study, and we have studied the impact of immobilization on the enzyme activity. So why the immobilization technique is necessary? Because we can use the that enzyme repeatedly, then it will be convenient for the product separation, and it can improve the enzyme stability also. So for that immobilization, we have tried the 3 different metrics, that is the gelatin, the sodium alginate, and combination of sodium alginate and the gelatin. We have used the wild type strain and the mutant strain for study of immobilization. Then again, we have carried out the comparative study of the different types of the matrices and the enzyme activity. And it was observed that the sodium alginate shows the promising enzyme activity. Then after selecting the sodium alginate as a carrier for the immobilization, we have optimized the different condition for the immobilization also. So Plackett-Berman design was also used for the statistical optimization. So factors we have chosen for this is the sodium alginate concentration, calcium chloride concentration, and the curing time. So amongst the 8 different trials, the 7th trial shows us the 46.05 units per ml predicted enzyme activity, while the 47.03 units per experimental enzyme activity we got, which is closer to that of the predicted enzyme activity. Then after studying the Pareto chart, it was observed that the significant factor, that is the factor which is having the p-value less than 0.05, is the calcium chloride concentration. So as only one factor is the significant from this study, so we have used the one factor analysis for the further studies, one factor analysis technique for the further studies. So only one factor we have chosen here, because only one factor shows the significant activity on the enzyme production, which is the calcium chloride concentration. After this one factor analysis, we found that this run number first, which is showing the 0.1 molar concentration of the calcium chloride, shows the promising enzyme activity. Then again, it shows the normal plot of the residues, which is the linear pattern, which verified the normality in the error term. That is, there was no any sign of the discrepancies in the data. Then the confirmation report we got with the help of the response surface methodology. In this particular result, we got that the prediction value and the validated value, the comparison between these. So predicted value is the 58.95 units per ml and the validated was 55.26, which shows the closeness to the predicted value. Then we have also carried out the characterization of the immobilized tannage of the wild type and the improved type of the stain. We have studied the effect of temperature, thermal stability, effect of pH, pH stability, and the kinetic values also, like the Km and the Vmax. So this is the result which shows the effect of temperature. The immobilized improved strain enzyme shows that the promising activity at the 40 degrees Celsius, and when we consider the stability of the enzyme, it is stable in the range of 40 to the 60. Here 85.32% activity retained by the wild strain, while at the 50 degrees Celsius, 90.13% activity retained by the improved strain at the 50 degrees Celsius. This is one advantageous point we got after the immobilization studies and the strain improvement studies. Then when we have studied the effect of the pH and the pH stability of the enzyme, it was observed that the optimum pH for the wild type of the strain and for the improved strain, for the improved strain, it was shifted to the pH 4. And when we check its pH stability, improved strain shows the stability in the range of 4.5 to the 7. So it will be the advantageous in the synthesis of the propyl gallate at the industrial level. Then the comparative kinetic study was carried out with the immobilized tannage produced by the wild strain and the improved strain of the Streptomyces variable. So increase in the Km value after immobilization may be partially due to the mass transfer resistance of the substrate into the immobilization medium. And it was observed that there is improvement into the Vmax of the enzyme due to the immobilization. It indicates that increase in catalytic efficiency or the turnover number of that improved immobilized enzyme. Then after this study, we have checked the operational stability of the immobilized tannage, and it was observed that we can use this immobilized enzyme repeatedly up to the 5 cycles, up to the 7 cycles we can use, because at the 7th cycle also it retains the enzyme activity 48.47%. Then the scale-up experiment was carried out. So gallic acid production was also studied, and the tannase activity was also studied for this scaling up reaction. For this purpose, we have used the production medium starting from the 15 to the 1500 ml, and it was observed that the tannase activity as well as the gallic acid concentration was remain constant, somewhat constant up to the 1500 level of the production medium. Then the FTIR analysis was carried out with the help of the effluent of the tannery industries. And this is the FTIR spectra of untreated effluent, which shows that various peaks was there and various types of the different groups are present. But when we compare this graph with this graph, this is the graph which showing the FTIR spectra of the enzyme treated effluent. It shows that there is a removal of the different types of the groups from this graph. Then we have studied the application of this tannase enzyme with respect to the debittering of the fruit juices. For that purpose, we have used the 3 different juices, amla juice, then the pomegranate juice and the orange juice. And effect of different concentration of the tannase was studied on the tannin degradation. So this was the different type of the concentration. This is the amount of the tannase was used and the amount of the tannin which get reduced from this level to this level with these 3 juices. Then the effect of tannase on the tannin degradation at different time interval was also carried out. And it was observed that as 120 minute, there is 75% decrease into the tannin content, as well as the 63% decrease and 69% decrease of the tannin content in amla juice, pomegranate juice, and orange juice respectively. So this picture shows us the amla juice, orange juice, and pomegranate juice before treatment, and this is the after treatment. This shows the haziness of the juice because of the tannin content. Then we have studied the application of this enzyme in the wine clarification. Again, the same method is used. The effect of different concentration of tannase on the tannin degradation was carried out. So it was observed that from this concentration amount of from this concentration of the tannin, it was reduced to this much concentration of the tannin when we use the different amount of the tannase. And the effect of tannase on the tannin degradation at different time interval was also carried out. So it was observed that after 10 minutes, the 50.25% amount of the tannin was remained from 98.8 microgram per ml of the tannin content. So this picture shows us the sample A and the sample B before the treatment, and this is the after treatment with the immobilized tannase. Then in the tea cream solubilization, we have also used this tannase enzyme. We know that after some time period on the surface of that tea, there is some haziness is formed, and that cream get accumulated. So we can remove that tea cream with the help of this enzyme, and it was observed that different incubation time, there is degradation into the tannic acid content from 198 microgram per ml to 120 microgram per ml when we treat it up to 5 hours. Then we have detected the presence of the gallic acid, whether the gallic acid is the major product into that fermentation broth or not. So, and it was observed that this is the detection of the standard gallic acid by HPLC and detection of the experimental gallic acid by HPLC. So it shows that the major peak of the gallic acid, and it is matching with the standard graph of the gallic acid by HPLC. So this result shows that after the fermentation, the major product into that fermentation broth is the gallic acid. So the conclusion, searching the natural source of the tannin that can be effectively utilized by the microbes, it is possible to design a process for the production of the tannase, which is more economical and environmental friendly. Many of the rare genera of actinomycetes have been neither explored nor manipulated for their biotechnological and industrial application. So with the help of this study, production and process optimization of actinomycetal tannase by using the statistical design offers the feasible approach for enhancing the tannase activity. 16S rDNA analysis of the actinomycete isolate was carried out, and the DNA sequence of actinomycete stain is submitted to gene bank with the accession number KY214403. The statistically optimized production medium resulted in 2-fold increase in the enzyme activity, while UV irradiation was found to be the effective to enhance the enzyme production. Purification with DEAE cellulose column chromatography resulted in 22.67-fold purification of the enzyme, and the purified enzyme showed 2 bands, one major band of molecular mass of about 150 kilodalton, and second band of molecular mass of about 66.4 kilodalton. Then impact of immobilization on tannase activity reveals that the immobilized enzyme of improved strain of Streptomyces variabilis is more efficient, and Plackett-Berman design and response surface methodology suggested the importance of calcium chloride concentration for tannase activity. Scale up of the formulated production medium from 50 ml to 1,500 ml was successful with no change in enzyme activity. The biocompatible removal of the tannin from tannary effluent using tannates produced by Streptomyces variabilis can prove an important tool. And enzymatic treatment of pomegranate, amla, and orange fruit juices to reduce the bitterness fruit to be an efficient tool for reduction of its tannin content. The result of enzymatic hydrolysis of tannin acid was detected by the HPLC analysis, which showed the appearance of N-gallic acid as a major product. So with this, I express my sincere thanks for giving me an opportunity to present my research.
Thank you, ma'am. Now the session is open for question and answering. If you have a question, please ask, ma'am. Or anyone to ask question, please raise their hand, or directly you can ask the question to ma'am.
There are some questions in chat box. What selective screening was carried out during initial stage of tannase producer? Selective
Yes, ma'am.
screening. Yes, we have carried out the selective screening. That selective screening means what? By using the tannin agar medium, we have taken the so many isolate on that tannin agar medium, and we have checked the whether it formed the zone of the hydrolysis on that plate or not. Then isn't it possible that every time this content will be changed for the same substrate? Isn't it possible that every time this content will be changed for the same substrate? The question I'm not getting.
Please repeat the question. Who has asked the question? Hello, if there is a no question.
Is it possible that every time this content will be changed for the same substrate? The substrate which we have used is the agro residues. So the tannin content of the agro residues vary depending on the geographical location from where you have taken that agro residues. Like the area, suppose Marathwada area, which is having the scarcity of the water. So in that, the secondary metabolite which are present into the plant, they are higher to remain in that particular environmental condition. So tannin content is higher in such type of the agro residues or substrate in which the environmental conditions are not feasible for their growth. So when we consider the Marathwada region, there is scarcity of the water and there is no availability of the water to that plant. So the plant is showing the higher accumulation of the tannin. So it depends upon the from which area, from which geographical area you have chosen your substrate.
Anyone want to ask the question? Please ask the question.
Tannase is intracellular or extracellular enzyme? Tannase is an extracellular enzyme. It is an extracellular enzyme. We have checked this with the comparison.
Ma'am.
Yes.
Your voice is not audible.
Hello, am I audible?
Yes, ma'am, you are audible.
Yes. Next question is tannase is intracellular or extracellular enzyme? So it is an extracellular enzyme. We have compared the intracellular activity and extracellular activity. Can you please explain how you have calculated the unit value for the enzyme? 1 unit per ml I have defined in the slide. That is the, you can refer that slide also. That is the 1 unit of enzyme is amount of unit of the gallic acid and glucose related per unit time.
Ma'am, also there is one question in chat box.
Respected presenter, ma'am, I have mini tab analysis problems. My research work, please, ma'am, give contact ID.
Okay.
You can take my email ID, sngirdhari@yahoo.com.
Okay, any question from the audience?
Good. Hello.
There is if any question in the audience? If there is no question, I
No
will
question
conclude.
from audience.
Okay. Prakash sir, ask your question. Prakash sir, you directly ask the question.
Yes. Yeah, actually, a very good study. I just wanted to know how much was the yield when you have mutagenized the culture. In
Yes,
other
sir.
words, how the enzyme yield was better compared to the wild type in a mutant. Because I don't see much difference as such.
Yes, sir.
Can you hear me?
Yes, sir. You are audible. What is your question?
Yeah, how much was the difference in yield or the activity of enzyme when you mutagenize the culture?
Yes. When we study the strain improvement technique, there is definitely the enzyme activity was increased with the strain improvement with the UV irradiation. The chemical mutagenesis not shows the promising activity, but the physical mutagenesis by UV mutation shows the promising enzyme activity with twofold increase.
Oh, that's all. But most of the results show that it is almost similar to the wild type. That is what actually was the major concern I saw in your slide.
No, sir. The results are not same with the wild type. Improved strain got the different result. There is shifting of the thermal stability. There is shifting of the pH stability. Also, there is the Km and max value is also showing the some changes. That's why we have chosen the improved strain for the further study.
Yeah, no. The thermal stability at 50 degree, I could see that there was an increase in the enzyme activity, but it should increase in other temperature also, right?
Yes. In other temperature, it was also increased, but it retains its activity. If you see,
Yeah, so how your mutant was better?
Yes, mutant was better when we consider that the thermal stability in the range of the temperature, that is the optimum temperature, and the when we see the range of the temperature with respect to the enzyme activity. So here we can say that the effect, when we compare the thermal stability, it can show the 53.12% the enzyme activity at 70 degrees Celsius. Also, it retains its 85.13% activity at 50 degrees Celsius also.
Oh, yes. With reference to usage of tannase, this can be used in a tannery effluent treatment also.
Yes,
Tannery
sir.
effluent from
Yes, sir.
leather.
We can use for the wastewater treatment of the tannery industry, because we can remove the tannin content of that wastewater of the tannery industry, leather industry.
Yeah, because right now it is mostly a chemical process, and that is actually
Yes. Right now it is a
hazardous.
burning issue that the leather industry is highly polluting industry, because it discharges the tannin into the water,
Exactly.
and that wastewater contain the high tannin content. So we have also checked the enzyme on the treatment of that tannery effluent also.
Yeah, because one of the research which is being carried out at CLRI Chennai.
Yes, sir. In the Chennai, this problem is this is the burning issue,
we
because
were actually a part of that project where.
Yes, sir. In the Tamil Nadu and in the Chennai.
Because most of the tanneries, what they have in the sea coast area.
Yes.
Exactly.
Yes, sir. We have also.
So very good work. Congratulations.
Thank you, sir. Thank you for your appreciation.
All the best.
Any question? Okay. I will. If there is no, I will conduct the vote of thanks. Should I conduct, ma'am?
Yes, sir. Yes, ma'am.
Good morning, all of you. I am Dr. Aarti Kamble. working as an assistant professor in Sadhguru Gadge Maharaj College, Karad. Today here I am to give vote of thanks for this speech. Ma'am, you have a very nice and applied research. Your lecture is a very informative to us and all the audience. They get fascinated to your research, ma'am.
Thank
It's
you.
a very nice lecture, ma'am.
Thank you.
I can appreciate the lecture. It is a very informative and nice lecture, and that your style of presentation is also too much better.
Thank you, ma'am. Thank you for appreciating.
My heart, thank you, ma'am, for your lecture, and my heartfelt thanks to all the audience and also who made this event possible. And also, I am very thankful to Microbiologist Society. I am very also thankful to Dr. A.M. Deshmukh, who give an opportunity to as a host of the program or as a convener of the program. And now I'm here to say that the session is over. Thank you. Thank you very much, all of you.
Thank you.